Okay, so welcome to this video. In this video what we're going to discuss is cyclic AMP signaling. Okay, now cyclic AMP is one of the most important intracellular messengers within the cell. Okay, so let me just run you through the structure for this video then. We're going to start off with a basic discussion of what cyclic AMP is and how it's produced from ATP. Then we'll discuss the adenylyl cyclase enzymes and we'll have a look at the structure of the plasma membrane adenylyl cyclases. Okay, then we'll discuss the GS cascades. We'll discuss G protein coupled receptors and how they can activate GS heterotrimeric G proteins, and then how the alpha subunit of GS heterotrimeric G proteins can then go on to activate adenylyl cyclase enzymes. Okay, then we'll mention forskolin, which is a drug that can do the same thing as GS and activate adenylyl cyclase enzymes, and we'll have a look at how that works. Then we'll look at the classic target for cyclic AMP, okay, which is protein kinase A, or the cyclic AMP dependent protein kinase. Okay, and we'll have a look at its structure and then what it's going to do once it's been activated. Okay, then what we'll do is discuss how the GS cyclic AMP protein kinase A cascade can be turned off. So we'll look at phosphodiesterase enzymes and protein serine threonine phosphatases. Then we'll move on to discussing some really important examples of the GS cascade. So we'll have a look at the effect of adrenaline on hepatocytes of the liver, skeletal muscle cells, and adipocytes, and we'll have a look at what protein kinase A actually does once it's been activated in those cells. Okay, and that will allow us to highlight a very important principle, which is that actually this same cascade occurring in different cells can cause very different effects in different cell types. Okay, after that what we'll discuss is the Kreb protein and how protein kinase A can phosphorylate the Kreb protein and how that can then lead to changes in gene expression within cells. Okay, after that we'll talk about other targets for cyclic AMP which are less well understood. So we'll talk about Epac, the exchange protein directly activated by cyclic AMP and what it does, and then we'll discuss some examples of cyclic nucleotide gated channels, and the examples that we'll look at are the HCN channels in the heart, which are responsible for the rhythmic generation of action potentials by the sinoatrial node. Okay, once we've done that, we'll discuss a very important concept that is emerging in all signaling fields now in biology. Okay, and that's the concept of microdomains and compartmentation of cyclic AMP signaling. Okay, and we'll discuss that at length later on. Then finally, what we'll move on to is other means by which the adenylyl cyclases can be regulated. So we'll look at the GI proteins and what they do to adenylyl cyclases. We'll look at the G beta gamma subunits of heterotrimeric G proteins and what they do, and then we'll look at how calcium signaling can interact with cyclic AMP signaling and regulate it. Okay, right. So let's begin by discussing the generation of cyclic AMP from ATP. So cyclic AMP is arguably the most important intracellular messenger within cells. In the 1960s, it was thought that it was the only intracellular messenger within cells. Then they discovered how important calcium was as well, and now calcium is gradually sort of rising up to the same sort of stature as cyclic AMP has. Okay, but cyclic AMP is still probably the most important intracellular messenger. Okay, so let's start by discussing how you can produce cyclic AMP from ATP. Okay, so let me draw a cartoon for ATP. So firstly, let me say what ATP actually stands for. So ATP stands for adenosine triphosphate. Okay, so to understand the structure of ATP, all we need to know is what the structure of adenosine is, and then we can just add 3 phosphate groups onto that to get adenosine triphosphate. So what does adenosine actually mean? What is adenosine? Well, adenosine means the organic base adenine okay, bound to a ribose sugar. So that's what adenosine actually means. It means adenine bound to ribose. So let's just draw a little cartoon for this. So the way I'll show ribose is as a pentagon, like so, with its 5th carbon coming off up here. So remember, ribose is a pentagon-shaped molecule. You have the oxygen right at the tip here, and then 4 carbons down here, and then you've got a 5th carbon coming off over here. Okay, right, and it's the sugar that's used in RNA. Remember, the sugar that's used in DNA is a slight variant of ribose called 2'-deoxyribose. Okay, but in adenosine, we don't want the sugar of DNA, we want the sugar of RNA. Okay, so here is ribose, and I'll colour ribose in green here. Okay, and now what we want to do is attach onto the ribose sugar an adenine organic base. Now I'm just going to denote the adenine organic base by a square with the letter A written inside it. Okay, so that's how I'm going to denote adenine there. Okay, so this is the adenine organic base here attached to our ribose sugar. Okay, and that molecule there now, adenine plus ribose, that is adenosine. Okay, now if we want to turn adenosine into adenosine triphosphate, all we have to do is add 3 phosphate groups onto it. Now we've added the adenine onto the 1st carbon of the ribose sugar. We're going to add the 3 phosphate groups now onto the whole adenosine molecule, onto the 5th carbon of the ribose sugar. Okay, so up here they're going to get added on. So I'm going to denote each phosphate group by a little blob, which I'll colour in purple. Okay, so these are the 3 phosphate groups of adenosine triphosphate 1, and then the second one is bound off that, and then the third one's bound off that. Okay, so there's my cartoon for what adenosine triphosphate looks like. Okay, and these phosphate groups of adenosine triphosphate, they all have names. Okay, so the one that's first off this 5th carbon here is called the alpha phosphate, the second one is called the beta phosphate, and the third one is called the gamma phosphate. Okay, so that's the adenosine triphosphate molecule. Now, how do you create cyclic AMP from adenosine triphosphate? So firstly, what does cyclic AMP stand for? Okay, well the C stands for cyclic, obviously, but what does the AMP stand for? It stands for adenosine, okay, that's the A, and then the MP is now for monophosphate. Okay, so if we were just talking about adenosine monophosphate, that would just be what we've drawn over here, the adenosine molecule, the ribose sugar with the adenine organic base attached to it, with the single alpha phosphate coming off it. That would be adenosine monophosphate, but we want cyclic adenosine monophosphate. So in fact what we're going to do is we're going to attach this alpha phosphate group onto the 3rd carbon of the ribose sugar here. Okay, we're going to break the bond between the alpha and the beta phosphate, and we're going to instead bind the alpha phosphate onto this 3rd carbon here, and that will create us another cycle in this structure. So let me now show you the structure of cyclic adenosine monophosphate. Okay, so here is our ribose sugar with our adenine bound to it here. And now we've got the alpha phosphate coming off the 5th carbon here, and it will be bound to now that 3rd carbon of the ribose sugar. So to colour this in, here's our ribose sugar in green. Okay, here is that alpha phosphate group that's now formed a new cycle here, and that's why it's called cyclic adenosine monophosphate. And then I'll colour in the adenine organic base again in red here. Okay, so understand the difference between adenosine monophosphate, AMP, and cyclic adenosine monophosphate. In AMP, the alpha phosphate wouldn't be attached to the 3rd carbon like that. Okay, adenosine monophosphate would literally just be cut the beta and the gamma phosphate off here, and then you've got adenosine monophosphate. To turn it into cyclic adenosine monophosphate, you have to then attach that alpha phosphate group now to the 3rd carbon of the ribose sugar to create that new cycle there, and that's why it's called cyclic adenosine monophosphate, or just CAMP for short. Okay, now, the other product then that you will produce in this reaction is just those 2 phosphate groups linked together. So you'll still have the beta phosphate and the gamma phosphate linked together like so. Okay, and when you've got 2 phosphate groups linked together in this way, it's called a pyrophosphate molecule. Okay, so the other product of this reaction, whereby you create cyclic AMP from ATP, is pyrophosphate. And for short, pyrophosphate is often abbreviated down to PP for pyrophosphate, and then people put a little I subscripted like that, in the same sort of spirit as when people show phosphate groups, they put an I in the subscript. It means inorganic. Okay, right. So we've got CAMP and PPI as the two products of this reaction. Okay, so that's how we can generate cyclic AMP molecules from ATP molecules. So we can make cyclic AMP, this really important intracellular messenger, from a molecule that we know we have a lot of in the cytoplasm of the cell. Okay, adenosine triphosphate is the foundational energy currency of cells. So all cells have a lot of adenosine triphosphate. Okay, so we can easily create cyclic AMP from a starting substance that we have a lot of. Okay, right, so let's now talk about the enzymes that actually catalyze this reaction, because this reaction doesn't occur spontaneously, okay, and the enzymes which catalyze the conversion of ATP into cyclic AMP are called adenylyl, oops, and I was about to write adenylyl again, adenylyl cyclase enzymes. Okay, you can also hear them called adenylate cyclase enzymes. Okay, so if you hear adenylate cyclase, it means exactly the same thing as adenylyl cyclase enzymes. And throughout this video, we will be abbreviating down adenylyl cyclase enzymes to AC enzymes, A for adenylyl and C for cyclase enzyme. Okay, right. So in humans, there are 10 adenylyl cyclase enzymes. Now, 9 of these are what are known as plasma membrane adenylyl cyclases. Okay, and these are abbreviated as PMACs occasionally. Okay, so what this stands for is plasma membrane adenylyl cyclases. Okay, and these 9 adenylyl cyclases are going to be integral membrane proteins within the plasma membrane. I apologize for that S. Okay, so they're going to be integral membrane proteins attached into the plasma membrane of the cell. Then finally, there is another one called the soluble adenylyl cyclase. So you do have one adenylyl cyclase which is just floating around in the cytoplasm, and this is called the soluble adenylyl cyclase. So that little S there stands for soluble. Okay, right. Now, we're not going to study the one soluble adenylyl cyclase at all, okay? Its regulation is very different from the plasma membrane adenylyl cyclases, and its expression is much more limited, okay? Very few cells have the soluble adenylyl cyclase, whereas loads of cells have the plasma membrane adenylyl cyclases. Okay, so we're going to chuck the soluble adenylyl cyclase away and just focus on the 9 plasma membrane adenylyl cyclases. Okay, so let me tell you the names for these 9 plasma membrane adenylyl cyclases. Okay, so they're given the numbers 1 to 9, and usually the numbers for adenylyl cyclases are written in Roman numerals. So adenylyl cyclase 1 will be like so, then you'll have adenylyl cyclase 2, and it will go all the way up to adenylyl cyclase 9, and the Roman numeral for 9 is 1x, like so. Okay, right. You have these 9 plasma membrane adenylyl cyclases just called adenylyl cyclase 1 to 9. Okay, let's now discuss the structure of these 9 plasma membrane adenylyl cyclases, because they all have pretty much the same structure. Of course, their primary amino acid sequences will all be different. They're all coded by different genes. Okay, but as far as their broad structural features are concerned, they're very, very similar. Okay, so I now want to discuss the structure of all of these 9 plasma membrane adenylyl cyclases. Okay, so I'll draw a picture of these enzymes implanted into the plasma membrane. Okay, so these 2 parallel lines here are going to represent the inner leaflet and outer leaflet of the plasma membrane. Okay, and now I'm going to draw my adenylyl cyclase implanted into the plasma membrane. Okay, so they are a single polypeptide. That's something very important to say. Okay, they're not multi-protein complexes. They are made up of a single polypeptide on its own. Okay, so I'm going to have the N-terminus and the C-terminus, and those are going to be the start and finish of the enzyme. Okay, so here's the amino terminus of our polypeptide, which is going to be our adenylyl cyclase, and it starts on the cytoplasmic side. So underneath here, this is the cytoplasm. Up here, this is the extracellular fluid. Okay, and this line will now represent the polypeptide, so this represents the polymer of amino acids. Okay, so it spans the membrane once here, okay, and that portion where it spans the membrane, that will actually be an alpha helix. Okay, now I haven't drawn it as an alpha helix because it makes the picture look messy, okay, but that would actually be a hydrophobic membrane spanning alpha helix. Okay, and it's not just going to span it once, it spans it a huge number of times, 12 in total. And these 12 membrane-spanning alpha helices are clustered into two clusters of 6 membrane-spanning alpha helices. So here's one of the first clusters of 6 membrane-spanning alpha helices. And this first cluster of 6 membrane-spanning alpha helices, this is known as TMD1, okay, for transmembrane domain 1. Okay, so all of those 6 membrane-spanning alpha helices that all cluster together there, together they are known as transmembrane domain 1 or TMD1. So this stands for transmembrane, that's the TM, okay, and then the D is for domain, and then it's transmembrane domain 1. Okay, then in this loop that you're going to have on the cytoplasmic side between TMD1, this first cluster of 6 membrane-spanning alpha helices, and TMD2, which is going to be the second cluster of 6 membrane-spanning alpha helices, you're going to have a very important domain, which I'm going to put in a box here. Okay, and this domain is known as the C1a domain. Okay, then you have a little portion here that also has a name. The portion following the C1a domain that's in a box, this portion here is called the C1b domain, and this overall loop between transmembrane domain 1 and transmembrane domain 2 is called the C1 loop. Okay, so here now this is transmembrane domain 2, this second cluster of 6 membrane spanning alpha helices, and this loop in between TMD1 and TMD2 is the C1 loop, and it's split into these two domains. One of them is absolutely really important, which is the one in the box, the C1a domain, and then there's also the C1b domain, which is also important for regulation. Okay, but the C1a domain is going to be part of the actual catalytic portion of the enzyme, so that's really, really important. Okay, so then we've got TMD2 here, which is another cluster of 6 membrane-spanning alpha helices here, and now we've had all of the membrane-spanning alpha helices that we're going to get. Okay, and after that what you're going to have is the C-terminal tail portion, which is known as the C2 domain, and this again has a really important domain here called the C2a domain, which is believed to be dimerized with the C1a domain even before the enzyme has been activated, and I'll discuss that in a bit more detail in a moment. And then you finish off with the C-terminal here, and the portion that's after the C2a domain is called the C2b domain. Okay, so this portion in pink here, this is the C2b domain. Again, that's important in regulating the activity of the adenylyl cyclase, but the really important bit is then C2a, which with C1a forms the catalytic site of the enzyme. Okay, so although the transmembrane domains look mightily impressive, okay, they are not the actual catalytic portion of these enzymes. The catalytic portion of the enzyme is the C1a, C2a dimer here. Okay, so this is the catalytic domain. Now, I need to say something quite important now, because there is something that is still very much so controversial here. Okay, it is controversial as to whether C1a and C2a are always dimerized. Okay, I would say that the growing opinion is that they are, basically. Okay, even before the enzyme has been activated, C1a and C2a are dimerized. That's the growing consensus. Okay, however, there are still people who will passionately argue that C1a and C2a are not dimerized until the enzyme is actually activated, and will argue that that's the way that the enzyme is activated, that you have to dimerize C1a and C2a together in order to actually get the active adenylyl cyclase, and the inactive adenylyl cyclase is when the C1a and the C2a are not dimerized together. However, I think the growing consensus now is that C1a and C2a are pre-dimerized in the inactive enzyme, and then to activate this catalytic domain, what you need to do is cause a conformational change, which then activates the C1a, C2a dimer. Okay, and that's the view that we're going to go along with in this video, but I should say that it is still controversial. It's not set in stone yet. Okay, right. I'll stress again then that C1a, C2a dimer here, this is the catalytic domain, this is the portion which takes ATP in and can turn it into cyclic AMP and pyrophosphate. And we think it's usually dimerized together like I've drawn even before it's been activated, i.e. the inactive adenylyl cyclase will be sitting in the plasma membrane like so. Okay, and to activate the catalytic domain, you need something to cause a conformational change in that C1a, C2a dimer, okay, which will then activate its catalytic activity, allow an ATP molecule to come in and be converted into a cyclic AMP molecule and a pyrophosphate molecule. Okay, right, so I think we'll call it there for this video. In the next video, what we'll talk about, firstly, is the different families of the 9 plasma membrane adenylyl cyclase enzymes, okay, because these 9 adenylyl cyclases are grouped into families, which is quite helpful for remembering what activates each one of them. Okay, so we'll start by discussing that, and then we'll go on to the classical GS cascade. But before I just finish this video, I'd like to put one more thing out there, okay, which is that this portion prior to transmembrane domain 1 here, that now is highlighted in yellow, that's known as the N-terminus, okay, or just abbreviated down to the NT here. So this is the N-terminus of the adenylyl cyclase. Okay, right, so see you in the next video.